Namaskaram. Now I am going to discuss about two chapters. One is the cell, second one is about biomolecules. First comes to the welcome to KNA X NPL season 2. So what is the strategy I am going to follow about the cell? So first of all, in the NEET exam, from the cell you will get 3 to 4 questions, okay. Anyhow, minimum guarantee 3 questions you will get. In some years you will get the 4 questions also. So 3 4s are 12 marks are guaranteed. So what is the strategy I am going to follow while I am discussing the cell is, so most of the students read the cell, like everybody having their own idea. So what is my strategy with regarding the cell is, which places are students show some small negligence, okay, that particular point I am going to elevate in this topic of the cell, okay, you understand now? Now I am going to start with the first question. The first question is related to the find the wrong one the following regarding the centromere or primary constriction. Here we need to find which one is a wrong. First of all, when you read is a small or 1 or 2% of the students having confusion between the terms. One is called as a centromere, is also called as a primary constriction. Second one is that centrioles, next one is a centrosome. The next one is a kinetochore. At any condition, don't get confused between the centromere, primary constriction, centriole or centrosome. So here thing is like, there is a one non-membranous organelle, that one we are calling here it is a, that one we are calling the non-membranous organelle, we are calling here it is a centriole. When these centriole, when it become a 2 centrioles, if they arrange, there is a non-membranous structure is forming is called as a centrosome. So centrosome consists of 2 centriole. It is a non-membranous organelle that I will discuss. The next one is related to the centromere. Centromere is also called as a primary constriction. So centromere where 2 sister chromatids are attached. So that is why don't confuse between these two. Now I am going for the question discussion. In this question what I place like, there are classification of the chromosomes based on the position of the centromere. As we know, first one is a metacentric chromosome. In some chromosomes, centromere are present in the center of the chromosome to give the 2 equal arms. Second one is called as a submetacentric, in which the centromere or primary constriction is slightly away from the center to give the 1 unequal arms. One is a long arm, another one is a short arm. Here we need to remember, long arm is represented, long arm and short arm are represented by P and Q arms. The next one, metacentric, followed by the submetacentric, followed by the acrocentric. Centromere is almost to the end, not to the end, almost to the end to give the extremely long and extremely short. Next, last one is called as the telocentric. Now first I am giving the confident is that based on the position of the centromere or primary constriction, 4 types of chromosomes. One is called as a metacentric, submetacentric, don't confuse between the acrocentric and telocentric. That's okay, that's very good. The next one is what you get missed, you know, when you are seeing the about third option, when you are seeing every, before that one clarity. Primary constriction associated with every chromosome. You need to understand, primary constriction or centromere associated with every chromosome, but secondary constriction associated with only some of the chromosomes only. Those are called as a satellite chromosome, satellite. That is why. Now, this here option C I am reading. Every chromosome have a centromere visible when the cell in the non-dividing state. It is not visible in the non-dividing. Centromere or primary constriction visible only when the cell in the dividing state. This point sometime you may miss. Be careful. I said this particular area, classification of the chromosome based on the centromere area. Almost cases lo there is a one question to ask. Now I am moving to the next question. The second question is related to the nice one. I connected between the cell and molecular biology. What is the exact location where, you carefully observe, 5.8 srRNA, 18 srRNA and 28 srRNA synthesis takes place. So how I correlated with molecular biology, you know, when you are reading this indicates itself. When I said this one, these 5.8 srRNA, 18 srRNA and 28 srRNA. First, when I said itself, this is belongs to eukaryotic cell. In eukaryote, first point I am giving clarity here. In eukaryotic cell, there are three types of RNA polymerases are present. For example, RNA polymerase 1, eukaryotic cell. RNA polymerase 1 involve the synthesis of 5.8 srRNA, 18 srRNA and 28 srRNA. RNA polymerase 2 involve the synthesis of hnRNA and mRNA. RNA polymerase 3 involve the synthesis of tRNA, 5 srRNA and small nuclear RNA. So with this point I said, okay, eukaryotic cell or eukaryotic nucleus consists of three types of RNA polymerases. One thing, when you are seeing 5. Second and last one, important one, RNA 5.8 srRNA, ribosomal RNA, 18 srRNA, ribosomal RNA, 28 srRNA, rRNA. In shortcut, if you want to ask where these rRNA synthesis takes place exactly, immediately you can go for answer is a nucleolus. mRNA synthesis takes place in the nucleus. tRNA synthesis takes place in the nucleus. rRNA synthesis also takes place in the nucleus, but rRNA synthesis takes place at a specific position called nucleolus. So that particular point followed by this particular 2 points are very important. Moving to the next question. So the next question is related to, in animal cell, careful, in animal cell, protein synthesis or translation occurs at, this is somewhat more standard to the normal person. Here protein synthesis or translation process means, when you are speaking about the animal cell means sir is speaking about the eukaryotic cell. Immediately translation process when it comes, immediately student will think at the rough endoplasmic reticulum bus. But protein synthesis is not only occurs at the rough endoplasmic reticulum. For example, the question comes, for example, in eukaryotic cell, if the question comes, in eukaryotic cell, where is the protein synthesis? In eukaryotic cell means what you need to clarity? Protista, fungi, plants, animals. If 4 kingdoms are belongs to eukaryotic cells. If you ask protein synthesis, what are the location where takes place in eukaryotic cell? First one you need to go for the RER. Second one is that at matrix of mitochondria. Next one is a stroma of the chloroplast. Up to here students are somewhat clear. But if you observe, there is a nuclear membrane is there, if you remember. Outer inner nuclear membrane and outer nuclear membrane. If you slowly observe under the nucleus, outer nuclear membrane is a continuous with a rough endoplasmic reticulum. There is a small word, 2 areas. One is at rough endoplasmic reticulum is there. Second one is a nucleus also they mentioned. people will get that particular point somewhat they will deviate. That is why outer nuclear membrane is a continuous with the rough endoplasmic reticulum, so that what you are speaking about the RR, same way outer nuclear membrane also associated with the 80S ribosome. That is why if you ask eukaryotic cell, protein synthesis or translation process occurs in the eukaryotic cell, RIR. There 80S ribosomes involved in the protein synthesis. Matrix of mitochondria, 70S ribosome. Stroma of chloroplast, 70S ribosome. Outer nuclear membrane, 80S ribosome. That is, if you take in animals only, in animals there is no chloroplast, na? That is why 3 location, RER, next one outer nuclear membrane, matrix of mitochondria. These 3 areas in animals, protein synthesis or translation process takes place. Going for option D, all of the above. The next one is related to this one. Find the incorrect statement about the eukaryotic cilia, flagella and centrosome. Okay? First option. Cilia and flagella are outgrowths of the cell membrane, that we will speak. Second option I am going. Cilia and flagella, eukaryotic cilia and flagella, characteristic feature is the presence of 9+2 arrangement. This is very important. Not only conceptual, but also exam point also important. Rare particular area. But I am telling eukaryotic cilia, eukaryotic flagella, characteristic feature is the presence of 9+2 arrangement. But in case of the prokaryotic cell, that flagella consists of just 3 portions. One is called as a basal body, followed by the hook, followed by the filament. Filament is made up of only one protein only. That protein is calling as a flagellin. So don't compare anything structure wise between the prokaryotic flagella and eukaryotic flagella. Third option. Centriole characteristic feature is the presence of 9+0 arrangement. That is also important. Next one, last option, nice option. Basal body of the eukaryotic cilia and flagella formed by the centrioles. This is a superb option D. Now comes to the what is the wrong in this one means whether you take the prokaryotic flagella or eukaryotic flagella. But for example, when you consider this one is a cell, when you consider this one is a cell, this is a cell membrane, always remember, if you want you can take this is a cell wall, just for understanding. Now always you take cilia or you take flagella, either you take prokaryote or eukaryote, this one is arising from only cell membrane. It's not arising from the cell wall. It is arising from the cell membrane. That is why whether you take prokaryote or eukaryote structure, cell bodies are there, basal body. Our basal bodies are always residing within the cell membrane. That is why I am telling. Cilia and flagella are outgrowths of cell wall Allah. They are outgrowths of cell membrane. Next one, that only I am stressing, just you can check your brain whether you are clear about this point or not and just leave it. Next time going forward. The next one is a find wrong one about the non-membranous organelle in a cell. Just first one, wrong one. Ribosomes, inclusion bodies, centrosome. Up to here I hope. So, what are the non-membranous organelles present in a cell? When you are telling, everybody may definitely remember the ribosome. Some persons will remember the centrosome. Up to here, everybody sure the person who is going for the NEET exam. But there are 4 non-membranous organelles present in a cell. One is a ribosome. Next one is the inclusion bodies. Next one is a centrosome. Next one is a nucleolus. There is a 4 non-membranous organelles. Now clarity. Ribosomes are non-membranous present in both prokaryotes and eukaryotes. Inclusion bodies are non-membranous present only in the prokaryotes, bacteria. Next one is a centrosome, non-membranous say, but present in the eukaryote, but in the eukaryote, centrosome not present in the protista, not present in the fungi, not present in the plants, present only in the animals. Next one is a nucleolus, non-membranous structure present in the eukaryotes. So that you should know the 4 non-membranous structure along with its clarity. Next one is ribosomes discovered by the George Palade. So we are calling as a Palade granules. Good. They are chemically, this point everybody will show some negligence. Here I am telling, ribosomes are chemically ribonucleoprotein. That means consists of different types of rRNAs and proteins. Last year I hope I remembered how many number of proteins associated with the ribosome. There are 80 types of proteins, 80 proteins associated with the ribosome. So that okay. Now option D I am reading. Neural control. Nasal granules are nothing but rough endoplasmic reticulum and free ribosome, ATS ribosomes, okay. So ATS ribosome, rough endoplasmic reticulum called nasal granules in case of neurons. When you are studying the what it called as a neuron structure, nasal granule, a typical organelle called as a nasal granule. Nasal granules are nothing but rough endoplasmic reticulum present in the cell body and dendrites, not present in the axons. So option 1, option B, option D are clear. Option C, just one time you can check. Prokaryotes containing a 70S. Eukaryotic cell containing 80S ribosomes only. Now it almost look like current to almost students, but prokaryotes containing 70S ribosomes correct, but eukaryotes containing 80S alla, eukaryotic cell containing both 70S and also 80S. Now I already said in eukaryotic cell 80S ribosomes are associated with the rough endoplasmic reticulum followed by the outer nuclear membrane. 70S ribosomes are present associated with the matrix of mitochondria and stroma of chloroplast. That is why eukaryotic cell contains 80S alla. Eukaryotic cell containing both 70S and 80S. While you are reading the option, read properly in the final exam. Going forward, the next one is related to the find the incorrect statement about the semi-autonomous organelles. There are 2 semi-autonomous organelles are present, mitochondria and chloroplast. We need to find incorrect. First you can look the question. Both organelles are the double membrane organelles. Yes, mitochondria consists of 2 membranes, chloroplast also consists of, but there is a difference. In case of chloroplast, outer membrane and inner membrane both are smooth. But in case of mitochondria, outer membrane is a smooth, but in case of mitochondria, inner membrane shows a certain foldings. Those foldings are calling as a cristae. You know why this inner mitochondrial membrane is a folded. You know not. You should know why inner mitochondrial membrane is a folded in the form of cristae. That's okay. Next option, both mitochondria and chloroplast containing a small, very nicely they wrote in the NCERT, small circular double-stranded DNA and 70 S ribosomes, correct, I discussed. Next option D I am telling, Krebs cycle occur in the matrix of mitochondria, clear. Electron transport chain or electron transport system occurring in the inner mitochondrial membrane, sure. Light reactions occurring in the thylakoid membrane, because thylakoid membrane only consists of light absorbing pigments in the form of photosystems. Next one is a CO2 fixation, or you can call it as a dark reactions occurring in the stroma of chloroplast locations. Just I am giving clarity to you because this is towards the tip of the exam. Now comes to the option C, a small logic I placed. Both organelles, mitochondria and chloroplast, containing enzymes for synthesis of the carbohydrates and protein. A small thing, maybe you will not notice there. First one you take the both mitochondria and chloroplast, both containing a DNA and both containing a 70S ribosome. When I said, sir, DNA is a present, DNA involves the transcription, mRNA. Ribosomes are present, mRNA undergo translation. So, both mitochondria and chloroplast shows the containing enzymes required for the protein synthesis. But, in case of chloroplast only, photosynthesis takes place. Photosynthesis results the formation of carbohydrates. That is why here both organelles you need to understand. Both organelles containing enzymes for synthesis of proteins. But the enzymes required for carbohydrate synthesis, the beautiful example is Rubisco. That enzymes required for synthesis of carbohydrates present only in the stroma of the chloroplast. That is why I am going for option C. Linearly you check, then you will understand. The next one is related to the C here. Find the wrong statement about endomembranous system. So option A, continuously they will ask in the different forms. Continuously ask in the different form. Endomembranous system include endoplasmic reticulum, Golgi complex, lysosomes and vacuole. Always asking question like, which of the following organelle not included under endomembranous system? This is so much frequently asked. So, 4 organelles because their functions are interconnected. Second option, RER involved in the synthesis of the proteins. Well known, superb statement. SCR, smooth endoplasmic reticulum, involves synthesis of lipids. Nice. And also involved the synthesis of steroid hormones. Here, this is a steroid hormone only in case of animals. So, these steroid hormones in our syllabus, when you go to the chemical coordination, last page. So, chemical classification of hormones there. So, 5 hormones only steroid. What is the meaning of steroid hormones? The hormones synthesizing from the cholesterol or cholesterol derived hormones are calling as a steroid hormone. Non-steroid hormone, the hormones are not synthesizing from the cholesterol. Only one example is a Saheli. That is why here I am speaking about the steroid hormone, just 5. Testosterone, estrogen, progesterone, aldosterone and cortisol. 5 only. T, E, P, A and C. Testosterone, estrogen, progesterone, aldosterone, cortisol. If 5 only comes under the steroid hormone, they are synthesizing from the cholesterol. Next I am going for the option C. The hydrolytic enzymes of the lysosomes are optically activating ACDPH. Nice one. STPH, if you remember, the pH is a 4.5 to 5 pH. Nice statement, beautiful statement. Last option. The tonoplast, this name also important. The membrane of the vacuole is called as a tonoplast. Allows the movement of ions and other materials along the concentration gradient. It is again as the constant. That means if you take this is a cell, assume. This one is a vacuole you can take. In the vacuole, concentration of solute higher than the cytoplasm. Automatically molecules are moving from low concentration to the high concentration. That is why this is not along the concentration gradient, that is again as the concentration gradient. Otherwise, this is not facilitated diffusion, that is active transport. That is why I am going for option D. I hope you are clear. Next I am going to the next one. Observe the statement about the Golgi complex. How many are correct? So see one by one. Consists of Golgi complex consists of many flat or disc shaped cisternae. There they gave us that diameter also they mentioned just one time. NCERT we should not neglect. First point correct. Next point is released. Cisterna are arranged near the nucleus. That's very good. Very close to the endoplasmic reticulum with distinct convex cis or forming phase. In 3 words people will get confused. And the second phase is calling as a concave, trans or maturation phase. This is a correct. So correct, you also need to think. Third one, the cis and trans phases are entirely same. No, the chemical nicely made. The enzyme composition of the cis phase is different from the enzyme composition of the trans phase. That is why here so small wrong. That is the same Allah. The enzyme composition of the cis phase and trans phase both are different, but they are interconnected with each other. So everything is correct except same. Understand? So this statement I am making it as a wrong. Next, fourth, nice one. Golgi complex mainly perform the packing of materials. Superb function. One more superb function is there. After packing the material to be delivered within the cell. Extraordinary function of Golgi complex. Delivering within the cell. Sometimes the proteins are sending out of the cell. That is that property is called as intracellular or intercellular protein targeting. So that function is in +2 level. They simply said the Golgi complex performing the packing of materials to be delivered intracellular within the cell or secreted outside. A nice point this is. The number of proteins synthesized at RER are modified in the cisternae of the Golgi complex. Yes, modify. What is that modification? We will see later. And released out, not from the cis phase, that is from the trans phase. You need to imagine the protein synthesized by the endoplasmic reticulum. The proteins synthesized by endoplasmic reticulum come and fuses with the cis phase. Once the modifications over, the matured protein, the functional proteins delivered out from the trans phase of the Golgi complex. So if you have imagination only, you can understand this point. This one is a wrong. This sixth one is a nice one. Okay? Golgi complex is a site for the formation of the glycoprotein and glycolipids. You need to understand. Proteins are associated with the carbohydrate, lipids are associated with the carbohydrate. That is called as a formation of the glycoproteins and formation of glycolipid. This every option is a superb. The functions of Golgi complex itself is a superb. That is why I am going for third and fifth one are wrong. I am going for option B. The next one is related to, see, ninth question I played something. If you observe, similar question in biotechnology chapter 2 times given. One time I am very sure. First observe this one. Question observe first. Find incorrect statement about lysosome. About lysosomes. So the question the last year or before last year NEET exam previous question is that find the incorrect statement about the endonucleases or related to the nucleases. Similar question I copied here in the different way. First option. Lysosomes are the membrane bound vascular structures formed from the process of packaging in the Golgi complex. Superb statement. Superb statement. So one, that's correct. So that is not only correct, that statement is related to the lysosomes. Next one. Containing hydrolytic enzymes are optimally at acidic pH. Correct. Already I said 4.5 to 5 pH. Nice one. Next one is, this is also correct with respect to of the lysosome. Option D one time you can check. Enzymes of lysosomes are capable of digesting carbohydrates. That is a carbohydrates. Proteases causes the breakdown of proteins. Lipases causes the breakdown of lipids. Nucleases causes the breakdown. So this is also correct with respect to of the lysosome. Correct. Option C just one time check. Vacuoles containing a water, sap, other excretory substances not useful for the cell. Exactly as the statement present in the cell under the vacuole. But this statement is also correct, but this is not a reference of the lysosome. You need to understand. A, B and D are both A, B, C, D all are correct statements only. But here we need to find incorrect statement with a reference of the lysosome. This option C is not related to the lysosome. Here clearly mentioned it is related to the vacuoles. So this is already I said, this is I inspired from the one of the previous year question given in the NEET exam. That is why I said. Now I am going next one. Read the following statement about the cell wall. Find the incorrect. Let's see. Almost everything is a statement wise because now you need to regain while you are seeing the video. We need to correct the incorrect statement. Okay. Cell wall is a rigid. That's good. Non-living. Absolutely. Cell membrane is a living. Cell wall is a non-living part. Present outside the cell membrane. Present in all fungi. That's very good. All plants, okay. All bacteria, okay. We'll speak about the first statement. Second option. Cell wall of bacteria is made up of peptidoglycan. Nice. So here up to here I am taking. Cell wall of the bacteria made up of peptidoglycan. It's a polysaccharide, especially heteropolysaccharide, okay. The enzyme, the cell wall of I am connecting with the biotechnology. Cell wall of bacteria made up of peptidoglycan. Opposite time done. The enzyme causes the breakdown of cell wall of bacteria. That enzyme is called as a lysozyme. That is why I can say this. Lysozyme is an enzyme causes the breakdown of cell wall of bacteria. Otherwise, the enzyme causes the breakdown of peptidoglycan is called as a lysozyme. Okay. Fungal cell wall made up of chitin. That's a superb. I will discuss again about the chitin. That's correct. The enzyme causes the breakdown of cell wall of fungi. It is called as a chitinase. If you observe in the isolation of DNA, I am comparing. Next one is algae cell wall made up of the cellulose, galactons, polymer of galactose, mannons, polymer of mannones, and calcium carbonate also. Okay, right. Plants are made up of cellulose, hemicellulose, pectins and protein. Indirectly, the enzyme causes the breakdown of the cell wall of the plant is called as a cellulase. Lysozyme, chitinase, cellulase. Okay, this statement correct. The next one is middle lamella is made up of calcium pectate. Correct. That word itself correct. Holds the diff. That's okay. The cell wall and middle lamellae transferred by the plasmodesmata. That is equals to the gap junctions in case of the animals, which responsible for the connection the cytoplasm of neighboring cell. Indirectly, this is responsible for the communication among the neighboring cells. Now comes to the first option. You can clearly understand. Cell wall is a rigid non-living structure present outside the cell membrane of all fungi. That's good. All plants, that's good. But all bacteria is wrong. That's because 99% of the bacteria having the cell wall. The only known bacteria without cell wall is called as a mycoplasma. That is why everything is exactly correct except all bacteria. You need to understand the small thing I applied. Now I am going forward. Which type of lipids? Okay, this is a good question. Okay. Which type of lipids mainly present in the cell membrane or plasma membrane? That's mainly. This I am going for the phospholipids. Phospholipid. Now I am telling a statement. Phospholipid. Plasma membrane also consists of cholesterol. Cholesterol is also belongs to the lipid. But here we mention is a mainly. That is why phospholipids are the main structural components of the plasma membrane. One more time. Plasma membrane made up of mainly one type of lipids are calling as a phospholipids. One. In our syllabus, only one phospholipid is present. If you remember, that is a lecithin. Lecithin. Don't compare with the lectin. Lecithin. So, lecithin is the only example of phospholipid present in our syllabus. Now, I am going forward. The next one is related to the, read the following statements about the eukaryotic cell, find the incorrect one. Okay. Eukaryotic cells include protista, fungi, plants and animals. Okay. I hope everybody clear. Make check whether your brain is correct or not the statements. What you are seeing, what you are spending half an hour or 1 hour in front of the video, that should be satisfied. Now, these cells contain, eukaryotic cells containing a extensive compartmentalization of the cytoplasm. Superb usage by the NCERT. Extensive compartmentalization of the cytoplasm, because eukaryotic cell cytoplasm consists of number of membrane bound organelles. That presence of membrane bound and organelles is called as a extensive compartmentalization. So, thank you the professor who wrote the NEET NCERT textbook. Next one is that containing cytoskeleton, well organized nucleus with nuclear membrane. Correct. See, last option. All eukaryotic cells are identical. Wrong. This is the wrong. All eukaryotic cells are not identical. When you compare, in NCERT they compared with the plant cells and animal cells. Plant cells having the cell wall, animal cells don't have the cell wall. Second one is the plant cells having the vacuole, animals don't have the vacuole. Most of the plants lacks the, what it called as a centriole centrosome, animals containing a centrosome. Just randomly they compared. Means because eukaryotic cell is not belongs to one organism, there belongs to 4 kingdoms. So that is why all eukaryotic cells are not identical. Basic structure is same but not same to same. That is the meaning of that. So I am going forward. The next one, find the correct statement about the cell membrane. Nice. Randomly I do. That statement I took just to overview. Chemical studies of the cell membrane. Especially the impressive one. Human RBC. That means most of the there is a small reason will be there. The small reason is there. Most of the research about the plasma membrane that occurred by taking the plasma membrane of only human RBC. One more time. Most of the research about the plasma membrane structure function, that is by taking only the plasma membrane of the human RBC only. There is a reason for that. Now, okay, decided this is the correct. A well accepted model for structure and function of the cell membrane. Superb fluid mosaic model proposed by the great Singer and Nicholson sir. Here also mentioned in the NCERT, somewhat careful. Cell membrane consists of, cell membrane of RBC consists of, this everybody remembered, just I made it intentionally wrong. RBC contains 40% of proteins, 40% of the lipids, 52% of the proteins. I hope you can understand. 52% of the proteins, 40% of lipid. Remaining 8% you will get out. Remaining 80% belongs to carbohydrates. So that is why this statement I made wrong. One more time while you are checking the video, just to check. Next one is that depending on the ease of extraction, the proteins of the plasma membrane, 2 types, peripheral proteins and integral protein. Nice one. Next one, an example of the active transport. If you remember, sodium potassium pump, or you can call it as a sodium potassium ATPase. How much this is plays an important role in maintaining the resting membrane potential in nervous system is extraordinary. That is why while I am speaking this one, where if it possible, you can read that sodium potassium pump function in the neuron. It is not only involved in the maintenance of resting membrane potential, but also it creates the negative charge to the cytoplasm. That 3 sodiums to send it out and 2 potassium send it inside per ATP. That point just one time careful. So I am going forward. So in this one all are correct except C. Going. The next one is related to the all are the functions of this type of questions are possible to ask in the simple way. All of the following functions of cytoskeleton except mechanical support to the cell. Superb. Motility, cytoplasm movement, that is called as a cyclosis, the viable property of the cell. Maintenance of the shape of the cell, that is also related to the mechanical support. Okay, so I am going for A, B, C are functions of cytoskeleton, don't neglect. I am going for option C. Now I am asking, so anyhow except answer C correct. Helps in the cell wall formation, helps in the DNA replication, helps in the respiration. It is whose function? This is a functions of mesosomes. Mesosomes are the characteristic feature of the prokaryote or characteristic features of the bacteria. Okay, I hope you are clear. So, going. Read carefully about the ribosomes and inclusion bodies in the prokaryotes and find how many are correct. Prokaryotes containing 70 S. What is the S stands for? S stands for, so many of the students are clear. So that S is stands for Swedberg unit or sedimentation coefficient. This is related to the density and the mass of the object or organelle. So that this one is a 30S smaller subunit followed by the 50S. Fundamentally required information. Nice. Correct. Polyribosome is a characteristic feature of the prokaryotes. There is a 2 words, polyribosome or polysome. Second one is a polycistronic. So these two are the something which is a characteristic feature of the prokaryotes. Several ribosomes, in brain 70S ribosomes associated with the 1 mRNA to make the translation is a more efficient. So this is also correct. Ribosome function, everybody will tell ribosomes are involved in the proteins and don't say like that. Ribosomes are site of protein synthesis. mRNA as a messenger during protein synthesis. tRNA is adapter during protein synthesis. Everything having its own role in the protein synthesis. Now, several, I make next one. Several reserve material in the prokaryote stored in the form of, just here small thing I change. It's not membranous, it's not membranous, this is a non-membranous, already mentioned. What are the 4 non-membranous structure? I am not repeating. So, I am making wrong. Inclusion bodies or inclusion granules include polyphosphate granules, synophysin granules, gas vacuoles, next one glycogen granules. 4 mentioned. Next one is the gas vacuoles are present in the purple green sulfur photosynthetic bacteria, most primitive bacteria. This is also correct. I am going all are correct except 4. I hope you can get it. So the total information of ribosomes and inclusion bodies I kept here. So no need to refer, just to overcome that one. Next one, cell envelope in the prokaryotic cell include all the except, easily find that is a ribosome, that is not the purpose. Here that one is a from outside to inside. First one is a glycocalyx outside, followed by the cell wall, followed by the cell membrane. If you want to make the inside like this, cell membrane, cell wall, followed by the glycocalyx. 3 layers are present in the cell envelope. Okay. Next one is cell envelope or plasma membrane. Small, everybody sure. That is related to the, some persons will get confused whether it is a semi-permeable or selective. Plasma membrane is absolutely belongs to selectively permeable. Plasma membrane, opposite I am telling. Plasma membrane or cell membrane never be Semi-permeable. That is why reasons later. Now this is not time for the reasons. I am making into confident and strong. The already knowledge present in your brain. So selectively plasma membrane of the prokaryote, selectively permeable. Plasma membrane of it is a selectively permeable only. How it selectively permeable? Let's see. Next one is, among the following, this is a question I took from the biological classification, okay? Among these one, which one shows the most extensive metabolic diversity? That is absolutely, it's not animals. Most extensive metabolic diversity observed in the bacterial cells are the prokaryotic cells. Nice question, okay? One time given question. That is I took and I placed it in the cell. The next one is, read the following statement about the cell theory and select the correct ones. We are going. I covered from the back side I started. So I reached almost towards the front. Schleiden, German botanist, observed the large number of plant cells. Observed all plants are made up of different types of cells. Okay, that's very good, correct. Schwanzer, British zoologist, studied the different types of animal cells. Reported all having the outer layer cell membrane. That's correct. Followed by the Schleiden and Schwanzer, they proposed the cell theory. Rodolphe Virchow modified the cell theory. Omnicellula, e-cellula. So, for your corresponding, so all statements are true. So, and that cell theory area I covered. So, if you receive and give last one, that one more thing I did for your, what did you call it as a, that for your understanding and this one. So, one time you can check. I hope this is a useful, what I remembered, so I hope it is a useful for all the students, okay. So I am telling, so it is not related to the question, okay. Singer and Nicholson sir is a famous for the fluid mosaic model of the cell membrane. Pallid, ribosomes. Fleming, the name chromatin was given by the Fleming sir. Golgi sir, okay. G.N. Ramachandran sir, triple helical structure of collagen. Triple helical structure of collagen. Okay. Stanley sir, crystallized TMV. Next one is a Diner, discovered the viroids, infectious nucleic acids containing a low molecular weight RNA without protein coat. Next one is a Whittaker sir, 5 kingdom classification. Artificial systems, system of classification given by the Linnaeus sir. Natural system of classification was given by the Bentham and Hooker. Next one, I enter into the photosynthesis. Priestley essentially discovered the essential role of the air in the growth of the green plants. If you remember, there is experiment, mint experiment. So that John, what do you call it, engine house, that sunlight is essential to the plant processes. Next one is Julius von Sachs, production of the glucose when plant grows. Next, Edelman, this is important. First action spectrum of the photosynthesis, not absorption spectrum. First action spectrum of the photosynthesis was given by the Edelman sir. Next one is related to the next one. Corollas von Neil, release of oxygen by the plants from the water but not CO2. Next one, great thing, almost peak level grade, that is a Dr. Peter Mitchell sir. He gave the chemiosmotic theory which explained the formation of ATP during both oxidative phosphorylation and also photophosphorylation. Dr. Peter Mitchell sir made the chemiosmotic theory. Blackman, photosynthesis last. There is a law of limiting factor when the reaction is controlled by more than one factor. That is, the rate of the reaction is determined by the factor which is present in the minimal concentration. So, Blackman's law of limiting factor. Next one is related to the Mishra sir. First isolated the nucleic acid, gave the name is called as a nuclein. Next one is a Messels and Stahl sir. They first proved, proved Semi-conservative model of replication in which organism? In prokaryotes, in bacteria, in E. coli. Next one is the Taylor and Co. Semi-conservative model proved in the eukaryote by taking the Vicia faba plant. Next one, T.H. Morgan sir, experimental proof for chromosomal theory of the inheritance. Don't get confused. Chromosomal theory of inheritance proposed by Sutton and Boveri. So, but experimental proof was given by the T.H. Morgan sir and discovery of the linkage. Next one, 2 times given question, Alfred Sustivant sir, gene linkage or genetic map. Based on the linkage, calculate the distance. Based on the distance, he arranged the genes arrangement. So that was not T.H. Morgan sir, that was Sturtevant. Hugo de Vries sir, Karl Correns and Eschermark rediscovered the Mendel laws. Sutton Boveri, already I said, they gave theoretical framework, chromosomal theory of inheritance. Henking sir discovered nuclear structure that play important role in the chromosomal sex determination. First he called it as X body, later we are calling as a X chromosome. I am making you to recollect. Jacob and Monan sir, very important. Discovery of lacoperon in the E. coli. Alex Jeffreys sir, so many times asked question. Alex Jeffreys sir initially developed that DNA fingerprinting. Sanger sir, molecular biology. First he sequences the protein, that is the insulin, and he developed the mechanism to sequence both proteins and nucleic acid. Rare breed on earth, Sanger sir. 2 times Nobel laureate. Next one is Alexander von Humboldt, great person. Species area relationship. Log S is equal to log C plus Z log A. Important area. Next one is Paul Ehrlich. Sir, revert to Popper hypothesis to explain how the different types of species are required to maintain the stability of ecosystem. Next one, the legendary Watson and Crick sirs, that is a double helical structure of bDNA, semi-conservative model of DNA replication, central dogma of life followed by adapter theory. So, I hope this one I made almost 31 discoveries. So, what I remembered, what are very important, I hope this one students will get some advantage. So, till now I said about the cell. So, from the cell, from the back side, I covered the nucleus, followed by I covered the cilia, flagella, centrosome, followed by I covered this one. So, from the back side, endomembranous system I covered, semi-autonomous organelles I covered, if you observe carefully. So, step by step I came in the questions, followed by I enter into the cell wall, then prokaryotic cell. So, like that, finally I came to the cell theory. Now, I am going for the biomolecules. Actually, biomolecule chapter is itself, it's a very big chapter now. So, I cannot make you to hear, but what are the areas? In the cell, I said the strategy I took is that which one you possibility to forget. But in case of biomolecules, the areas I will tell you. So, which areas are essential? That I made as a question to recollect. Okay. Now, I am entering the first question. And what is the strategy we are following before going to explain something is very important. So, find the wrong statement about the cofactor of the enzymes. Here I am telling. When you are speaking last one cofactor, there is more possibility of asking about the cofactor. When you are speaking about the cofactors, some of the enzymes requires non-protein parts are calling as a cofactor. 3 types. One is called as the prosthetic group, next one is a coenzyme, next one is a metal ions. I hope you are clear. The difference is very important. Prosthetic group is organic cofactor which is tightly associated with apoenzyme. Only example, heme is the prosthetic group of catalases and peroxidases. Definition should be very clear in your brain with one example. Second one is that these are also coenzymes, also organic cofactor, but they are loosely or transiently associated with apoenzyme. Next one, example. So, one more point, important point is there. Many of the coenzyme containing essential components are vitamins. Only example given, NAD plus NADP plus coenzyme containing vitamin E is niacin, vitamin B3. Next one is a metal ion. Metal ion are inorganic, exactly like a prosthetic group. But only different, prosthetic group is organic, metal ion is basically inorganic. So that only one example, zinc. Zinc is a metal ion required for the enzyme carboxypeptidase. So this is the description you need. So somewhat careful about while you are reading that area. Last option I am taking here. Catalytic activity remains the same when cofactor is removed. That is wrong. That enzyme lost its catalytic activity when the cofactor get dissociated. Whether you can take prosthetic group, whether you take the coenzyme, or whether you take a metal ion. Once the cofactor is dissociated from it, automatically enzyme cannot carry the biochemical reaction. So I hope you are clear with that area. That area how I am making is important. Next one I am going. The following reaction is mediated by which class of enzyme? Before that, which class of enzymes? There is a here you need to remember 6 classes of enzymes. Here that classes should remember in the same order. So I am telling OTHLIL. So O for oxidoreductases, class 1. T for transferases, class 2. H for hydrolases, class 3. L for lyases, class 4. I for isomerases, class 5. Class 6 is belongs to ligases in the same order with just general reaction. If you remember, that's very good. Okay, there is not cofactors are more chance to ask, but we should not neglect this. General reaction is important. The class is important. That general reaction important. Now I am taking this one is related to the, here the very simple, this one is a substrate, this is also substrate S prime. Here group is a G, some group other than the hydrogen. Hydrogen if it comes automatically the enzymes are belongs to oxidoreductases or dehydrogenases. Clearly mentioned in the NCERT as a definition. So group G is transferring from S to S prime. So mediated by enzyme, a specific class of enzyme belongs to transferases. I said 6 classes of enzymes, one time you can check. And their general equations check. That's enough. Next I am going forward. Competitive, superb question. And it is not about the question, superb concept. Competitive enzyme inhibition. In the competitive enzyme inhibition, substrate and inhibitor. For example, if you take like this consider as an enzyme. This is the substrate, this is the competitive inhibitor. So, substrate in the competitive inhibition, substrate and inhibitor are structurally similar. Don't call it as a structurally same. Both are structurally similar. That is in competitive. What is the reason for competition in competitive enzyme inhibition is the molecular shape of the substrate and inhibitor both are similar. This is a one. This is out of NCERT but asked one. During the competitive inhibition, Km value increases. Km value is nothing but substrate concentration. Very potential word. While you are reading the photorespiration, you will get that area. So that Km value is increases, but Vmax is also remains the same. Correct statement. Next one, the classic example of the competitive enzyme inhibition. The classic example, only example. Malonate is a competitive inhibitor of enzyme succinate dehydrogenase. If you remember, succinate dehydrogenase is an enzyme involved in the Krebs cycle. That is a converting the succinate to fumarate. One, this is a mediated by succinate dehydrogenase, involves the conversion of Fa, involves the synthesis of FADH2. That is how much important. So I am telling, succinate dehydrogenase is an enzyme involved in the Krebs cycle. That's everybody remember. But the same enzyme is also involved in the electron transport system as a complex tube. There is a very big logic is present between this one. Now I am telling the example is very important. Malonate is a competitive inhibitor of enzyme succinate dehydro. These three statements are very important. I am going for option 4. This area, cofactors area 1 is important, followed by the competitive inhibition is important. Conceptually important, simultaneous exam point of view also important. Rare combinations. Find the correct statement about factors affecting enzymatic activity. Okay, normally. Each enzyme shows the highest activity at a particular temperature and pH called as a optimal temperature and optimal pH. Okay, that's good. At low temperature, enzymes are inactive. Nice statement. This is application for the cryopreservation. Cryopreservation comes under the extensive ex situ conservation. So that's a nice one. At low temperature, enzymes are become inactive. At a high temperature, enzymes undergo denaturation. Denaturation. Because enzymes are nothing but proteins, nana. They loses the tertiary structure automatically. Okay, that's good one. Good statement. Next, enzyme shows the maximal velocity at a saturation point of substrate. This is also nice. The enzyme showing the maximal velocity where at the saturation point. All of the above. Considerably important, that particular area. But already I said, there is a Vmax remains the same, Km value increases. That competitive enzyme inhibition, this one is both are interconnected with each other. That is why I said this don't neglect but important. Now I am going forward. The next one related, find incorrect statement about the enzymes. Let's see. An enzyme like a protein, enzymes like a protein, actually enzymes are nothing but proteins. Has a primary structure, secondary structure, tertiary structure, and normally only. Okay, that's good. Third option I am going. Inorganic catalysts work efficiently at a high temperature and pressure. This is nice. But enzymes are functioning at our physiological conditions. Means in our body there is a 37 degrees centigrade only. But because that is why enzymes get denatured at a high temperature. That's correct. Fourth statement, important. Enzymes of thermophiles. What is the meaning of thermophiles? The organism, especially bacteria. In our syllabus especially belongs to archaebacteria. The enzymes of thermophiles are heat stable. Indirectly, the enzymes of thermophiles will not undergo denaturation at a high temperature. That is called as a, not to call it as a heat sensitive, you need to call it as a heat stable. When I said the enzymes of thermophiles are heat stable or thermostable, means immediately you can go for enzyme is a Taq polymerase. If you remember, very important, thermus aquaticus polymerase used in PCR, polymerase chain reaction. Nice, that is I said, this statement nice. Next, 99% of the enzymes are belongs, 99% of the enzymes are belongs to proteins, and no doubt about it. But some RNA, don't call it as a RNAs. Some RNAs only acts as a enzymatic activity. That RNA molecule acting as enzymatic activity called as ribozyme. So immediately in our total syllabus +1 +2, only one example of ribozyme. If you remember 23SR RNA, that 23SR RNA. 23SR RNA responsible for the formation of peptide bond. That is important. Plus, this is only in the prokaryotes or bacteria. So, 23SR RNA is an example of ribozyme. This enzyme responsible for the formation of the peptide bond in bacteria. That is important. So, this among these one, I am going for the NEET exam point of view. This fourth statement is absolutely stunningly important. Next one is that B I am taking as incorrect. Active site of the enzymes as nothing but functional form. All proteins with reference of the +2. All the proteins function. Tertiary structure is absolutely required for the biological activity of the protein. That is a given question. That is a given statement. Otherwise, tertiary structure is essential for the functioning of the protein. That is why active site of the enzyme is not because of the tertiary structure, is because of the, not because of secondary structure, it is because of tertiary structure. So, I am going. So, in this question, option D. Next one. Which of the following bonds? Superb question. Superb question. This is not the question, the way of their asking is different. Which of the following are examples of the dehydration? First one, I am taking all of the above answer. Thing is that glycosidic bond, the bond which connect sugars together. When you are telling, sir, there is a glycosidic bond present between nitrogen base and sugar, N-glycosidic bond, same to same. So, in general, glycosidic bond is a bond which is connecting the sugars together. Either you take disaccharide or polysaccharides. Next one is a peptide bond, the bond which is linking the amino acids together in the protein. Phosphodiester bond, which is linking the nucleotides together in a nucleic acid or polynucleotide chain. So that the glycosidic bond linking the sugars together, peptide bond linking the amino acids together, phosphodiester bond linking the nucleotides together. So that all are important. This is a nice question. But last year, before year, I hope I remembered. The given question is that glucose formula C6H12O6. If you remember, you did the question. Same question, way of asking is nice. Glucose C6H12O6. What is the molecular formula of the maltose, if you remember? Okay, one more application I will tell you. With regarding this one, you take any glycosidic bond, peptide bond or phosphodiester bond if you take. For example, if they ask, how many number of glycosidic bond, or I will take one example, peptide bond, I will tell you. You apply to the same concept. How many number of peptide bonds are present in the decapeptide? Decapeptide means a peptide made up of 10 amino acids, not 10 peptide bonds. So that how many number of peptide bonds are present in the decapeptide? Immediately you go for n-1. So decapeptide consists of 10 amino acids. How many number of peptide bonds? 9. Same thing you can apply for the glycosidic bond and also for example, if I say, if a protein is made up of 51 number of amino acids, immediately number of peptide bonds are 50. How many water molecules comes out? 50 water molecules. How many number of phosphodiester bonds present in a nucleic acid made up of 100 nucleotides? 99. That is why always number of peptide bonds, number of phosphodiester bonds, number of glycosidic bonds in that particular polysaccharide is a n-1. The way of asking is important. Now, the first and last amino acid, this is a well known, you expose to this question number of times. Those who are going for the next NEET exam, question, number of questions you did. So here last one as a, from point of us, last version. When you are taking this one, consider as a straight line. If you consider this is a polysaccharide. This is a first monosaccharide, last monosaccharide. First monosaccharide, last. First monosaccharide, if you take polysaccharide. If you take this one as a protein, if you take polysaccharide, first monosaccharide, last monosaccharide. If you take protein, this is the first amino acid, the last amino acid. If you take the nucleic acid, this is the first nucleotide, this is last nucleotide. Everything is the same except the frame of reference. So whether you are speaking about the polysaccharide or you are speaking about the polypeptide or you are speaking about the nucleic acid. If you take polysaccharide, left end is a non-reducing, right end is a reducing. If you take the protein polymer, left end is a amino terminal or N terminal, right end is a Cr carboxy terminal. If you take the nucleic acid, polynucleotide chain, molecular biology, left end is a 5' phosphate end, right end is a 3' hydroxyl end. So somewhat, I am giving the clarity. Such options will come in the statement wise questions. That is why I am going for option B. Okay, amino group is at the left, carboxyl group is at the right. The next one is related to the well known. Alpha helix and beta sheets are examples of primary structure, secondary. So I am going for secondary structure. Alpha helix, beta sheet are examples of the secondary structure. There one important point is there. Helix two types, right-handed and left-handed. But in the cell, only right-handed helix. Only right-handed helix are present in the cell. That is why. Okay. So that is a why. So somewhat a structure of protein is somewhat area. So I am more number, some of the questions I am giving in the structure of the protein. What is the functional form of hemoglobin? Here I mentioned A. A means adult. So here is that adult, this functional form. When you are speaking about hemoglobin, there is a 4 polypeptide chain including alpha to beta 2 alpha polypeptide chains to beta polypeptide chains. That means this protein consists of more than one polypeptide chain. Okay, one. This one is each polypeptide chain. So, this first point consider. Second point, each polypeptide chain consists of one heme group. One heme group containing a one ion. Each ion carries a one molecular oxygen. So, one hemoglobin molecules carries maximum of 4 molecules of oxygen. Second point consider. Without hemoglobin, 2 alpha polypeptide, 2 beta polypeptide. In sickle cell anemia, which polypeptide chain is affecting? Beta. At the 6th position, glutamic acid is replaced by the valine at the 6th position of beta polypeptide chain. So, sickle cell anemia. Next one is the 4th point relates to thalassemia. So, in thalassemia, sickle cell anemia is because of the substitution point mutation. Thalassemia is due to the deletion mutation. Sickle cell anemia belongs to qualitative anemia. Thalassemia belongs to quantitative anemia. Last one, because it consists of more than one polypeptide chain, the functional form of hemoglobin, adults, belongs to quaternary structure. See, if you don't know clarity about the hemoglobin, there is a conceptual error in your brain. That is why whenever you are speaking about the hemoglobin in adults, 4 polypeptide chains to alpha to beta. Next one, each hemoglobin molecule carries a 4 molecular oxygens. Next one, sickle cell anemia affected polypeptide chain is not alpha, it is a beta at the 6th position of beta polypeptide chain. Next one, thalassemia, it is also anemia, but sickle cell anemia is a qualitative anemia, thalassemia is a quantitative. In our syllabus, only example of quaternary structure which is a functional form of the protein is a hemoglobin only mentioned. There are certain proteins, but the mentioned one is a hemoglobin. So, I am going for this question. Next one. Identify the correct statement about the polysaccharides. Starch, and I gave the certain information which is useful to you. Starch, glycogen, cellulose, inulin and chitin are homopolysaccharides. In our biology, biomolecules, only homopolysaccharide only mentioned. Only 5 homopolysaccharides. One is a starch. Always polysaccharide should start with a starch because it is a representative of polysaccharides. Starch followed by glycogen. Next one is the cellulose, chitin and inulin. Among these one, starch, glycogen, cellulose are the polymers of glucose. Nice. Inulin is a polymer of fructose. Nice one, correct. Among starch, glycogen and inulin are stored polysaccharide. Chitin and cellulose are belongs to structural cell wall polysaccharide. I hope you are clear with my options. Chitin, this is a stunningly important 4th one. Chitin is a polymer of NAG, N-acetyl glucosamine, nitrogen containing polysaccharide. It forms the exoskeleton of invertebrates, arthropoda, and also forms the cell wall of fungi. So, among the animals, out of every 10 animals, 7 animals are belongs to insects. The reason, the most diversity of the insects is due to the presence of their exoskeleton made up of chitin. Biodiversity connecting point. In ecosystem, there is another point. Chitin is one of the most resistant substance to the organic decomposition. That is why this fourth statement is a. Next one is a cellulose. 4-1 correct. Just here I made. So here conclusion point. Starch gives blue color with iodine because starch consists of complex helical secondary structure. Complex helical structure. Cellulose will not give the blue color with iodine because of lack of helical structure. That is why I am going for fifth one is a wrong. So all are correct statement except 5. Now, so in this question number 10, all polysaccharide area completed. So no need to recover again if you listen properly. Find the mismatch is of the protein's function. Trypsin is an enzyme, correct. Collagen, already I mentioned collagen, 2 points I will tell you. Enzymes position I place trypsin na. One more enzyme is a rubisco. So ribulose 1,5-bisphosphate carboxylase or oxygenase. The most abundant protein in the world or most abundant protein in the total biosphere. Next one, collagen. It is a very important protein in the ground substance, that is extracellular matrix, connective tissue. Most abundant protein in the biosphere, rubisco. Most abundant protein in the animals, only collagen. Triple helical structure of collagen was given by the G.N. Ramachandran sir. So that this is a correct. Next, antibody. If you remember, IgG-A, IgG, IgM, IgA, IgD and IgE. So, infectious antibodies. GLUT4, it important, enables glucose. So, A, B, D, E correct. This is not insulin. That is not inulin, that is a insulin. While you are reading this one, don't confuse between the inulin and insulin. Okay, inulin is a polysaccharide, insulin is a protein. That is why this I made it wrong. So, intentionally to make the difference between the inulin and insulin. Now, find the mismatch of the biomolecules with the average composition of the cell. Proteins. This one, water is belongs to 70 to 90%. Average, okay, not absolute. Proteins 10 to 15%. Nucleic acid, this is a wrong, 5 to 7%. Carbohydrates 3%, lipids 3%. Ions are belongs to 1%. Chemical composition, okay. So if the question comes, what is the most abundant component in the cell? Water. What is the most abundant organic component? Proteins. Most abundant component water. Most abundant organic component is a protein. So don't get confused, that table is important. Last questions I gave the clarity among the functions of the proteins, okay. Next one is related to, there is a absolute every year given question, secondary metabolites, no doubt about it. So I gave the question, I covered the all in that questions. Questions is not like this, but people will get neglect. That is why every year there is a constant question on the secondary metabolites. That is why I am telling here. Alkaloids. What are the examples of alkaloids? Alkaloids include morphine and codeine. Next one is the pigments, carotenoids and anthocyanins. By closing eyes, you need to tell the all that particular table in the biomolecules. Lectin, not lecithin. Lectin example is a concanavalin A. I said don't get confused between the insulin and inulin. Don't get confused between the lectin and lecithin. Lecithin is a primary metabolite. Lectin is a secondary metabolite. Example is a concanavalin A. Essential oils, lemongrass oil. Toxin, abrin and ricin or ricin. Next one is the drugs, vinblastine and curcumin. Polymeric substances, rubber, gums and cellulose. Terpenoids, monoterpenes and diterpenes. Antibiotic, penicillin, streptomycin as you comfort. So I don't know what you will do. There is every year one question will come on the secondary metabolites while you are open that seal, while you are doing the questions. So just to remember, there will be question, there should be question on the secondary metabolites because they are commercially also such a important terms. For example, antibiotics. That is why I am not making varied. Triple star question, that is the secondary metabolites. Now next one. Amino acids are, this is I made intentionally to connect with the evolution. Amino acids are substituted methane. If you remember CH4 in the Miller's experiment, in the Miller, this is the first one. Amino acids are substituted methane. General structure is required. At least knowledge purpose. May not be asked, but at least knowledge purpose. In that area, just you consider the properties of the amino acids are amino group, carboxyl group, R group, like the statement. Zwitterion I didn't cover. One time you can check the zwitterion. At the different pH solution, the amino acid shows a different structure. So that here the connecting point is the evolution. In the Miller's experiment to prove the chemical evolution. So Miller took the methane, ammonia and H2. So at the temperature 800 degrees Celsius, when he took the methane, results the formation of amino acids because amino acids are substituted methane. So there is a link between these two, that is why I gave this question, okay? Not absolutely important, but conceptually, knowledge wise important. The next one is find the wrong one about the lipids, that lipid area. So people get somewhat, they will forget like mixing, everything is a mixing area, that paragraph. Generally lipids are water insoluble, correct? Lipids are a word that is generally water insoluble. Generally, that's nicely they used. R group, normally here we are using RCOOH. R group is extended from methyl group up to the 19 carbons. Pure NCERT, correct. Chemically, glycerol is a trihydroxypropane, correct. CH2OH, CHOH, CH2OH, the backbone of many of the fats, many of the lipids, sorry. Palmitic acid, in our syllabus given only 2 fatty acid. Palmitic acid is a 16:0, means 16 carbon atoms, including the carboxyl group, Arachidic acid alla, arachidonic acid. It contains 20 carbon, 20:4. 20, it's a polyunsaturated, 20:4, polyunsaturated fatty acids. That is why including the carboxyl group. Intentionally I made excluding the carboxyl group because you may skip. That is why you need to remember the only 2 fatty acids. One is a palmitic acid, 16 carbons, arachidonic acid, 20 carbon atoms. That including the carbon. What is importance of mentioning arachidonic acid behind this one? Arachidonic acid involved in the synthesis of prostaglandins. You may have heard. So the person who made this one, prostaglandin, during the parturition time, uterine contraction is not only involvement of oxytocin but also involvement of prostaglandins. That is why muscle, one more time, during parturition, uterine muscle contraction, smooth muscle contraction, synergic function of both oxytocin and also prostaglandin. That prostaglandins are synthesizing from the fatty acid belongs to arachidonic acid, which consists of 20 carbon atoms including the carboxyl group. It is an example of polyunsaturated fatty acid. Next one, that statement is correct. Next one, here this statement I made wrong because I wrote exclude. Next statement they asked one, okay, ginger oil they asked within brackets low there. So ginger oils having the lower melting point when compared with the fat. So that this statement, lipids area, one time you can check. So almost that information I covered, okay. The next one is real. Following biomolecules are present in the acid insoluble fraction. Acid insoluble fraction in general, they containing a biomacromolecules. Biomacromolecules molecular weight greater than 800 daltons. That include polysaccharides. Next one is the proteins. Next one is the nucleic acids. Next one is the lipids. Lipids means you should take only phospholipids only you should consider. These polysaccharides, proteins, nucleic acids, lipids, those are present in the acid insoluble fraction. Sorry. They are present in Karthik sir. Sorry, yeah. Karthik sir. Sorry. Karthik sir. 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 Adi slide down. Sorry, yeah. That's it. I understand. Okay, I will tell you that one. Thank you. That one it went. Where, sir? Okay. The last one. No, this. This almost. So this one. So last version is this graph is related to the mechanism of enzyme action. So this graph is very important. Okay. So 1 or 2 times already given question from this one. Okay. So I am giving the double star to this one, very important. Okay. That representations are very important. Okay. So anyhow, those who prepared already. So now today is 30th. So May 5th will be the exam. Okay. Those who prepared for everyone. So those who are writing NEET exam, those did hard work. So all the best. Okay. So thank you very much.